बिस्मिल्लाहिर्रहमानिर्रहीम। डियर स्टूडेंट्स, आज की जो वीडियो और लेसन है वो हमारा बायोलॉजिकल मॉलिक्यूल्स पे है। इसकी वजह ये बनी कि जब मैंने बायो एनर्जेटिक्स ये आपके सिलेबस में से एमडी कैट के सिलेबस में से लिया, तो उसमें जो रेस्पिरेशन का पोर्शन था उसमें जो मेरे ख्याल में एक थोड़ा पॉइंट जो डिस्कस करने वाला था वो था अबाउट द role of protein and fats in respiration along with the glucose. So I thought कि it may cause some difficulty for the students, तो इसलिए मैं इसको थोड़ा इलेबोरेट कर लूँ या थोड़ा अपने पॉइंट ऑफ व्यू से बता लूँ। उसमें एक तो क्यूरीज आई स्टूडेंट्स की कि जी इसी को हमें कंटिन्यू कराया जाए इन टॉपिक्स को। दूसरा उसके व्यूज़ थे ज़्यादा, तो मेरे ख्याल में स्टूडेंट्स को उसमें कुछ चाह रहे थे। तो फिर मैंने नेक्स्ट जो अपनी स्ट्रेटजी से तो मैंने ये देखा कि जो डिफ़िकल्ट टॉपिक हैं उनको डिस्कस करूँ मैं, या जिनमें थोड़ा इनपुट चाहिए। तो अदरवाइज अब बायोलॉजिकल मॉलिक्यूल्स भी अगर उसके साथ कंटिन्यूटी में करने हैं तो मैंने सोचा कि क्यों ना हम उसको भी ले आएं। तो जो यूनिट 3 है, इट कंसिस्ट ऑफ different subtopics. Number one is biological molecules. So biological molecules में भी हमें we are supposed to define and classify biological molecules and then to discuss the importance of biological molecules. ये हमारे सिलेबस में है। इसको आज एक जो वीडियो अभी मैं बनाने जा रहा हूँ या हम बात करने जा रहे हैं लेसन में, वो इन्हीं दो टॉपिक्स को कवर कर रही है। तो अब ये थोड़ी सी लेंथी हो सकती है क्योंकि इसमें तकरीबन सारे चीज़ों को डिस्कस करना होगा, आल्दो ब्रीफली, क्योंकि इनके इंडिपेंडेंट टॉपिक्स भी आपके सिलेबस में हैं। तो नंबर वन इज डिफाइन एंड क्लासिफाई बायोलॉजिकल मॉलिक्यूल्स, नंबर टू डिस्कस द इंपॉर्टेंस ऑफ बायोलॉजिकल मॉलिक्यूल्स. So, what is the precise definition of biological molecules? A molecule created by living organism and that is necessary for one or more generally biological functions in is referred to as biomolecules or biological molecules. Biomolecules comprise small molecules like vitamins, hormones, as well as massive macromolecules including proteins, carbohydrates, lipids, and nucleic acids. So, when scientists talk about biological molecules, they usually means organic compounds, mean the molecules built primarily from carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. So these include carbohydrates, for example, glucose, starch, lipids, uh, fats and oils are included, proteins, enzymes, collagen, and other proteins as well. We will discuss later on. Nucleic acid, DNA, and RNA. So all these molecules have carbon backbone and specific function tied to life processes. That's why they are named as biological molecules. Water is crucial for life, but it is an inorganic compound. So it is not considered a biological molecule like carbohydrates, proteins, lipids, and nucleic acid, but it is biologically very important. There is an independent portion of your syllabus. We will discuss it later on. Carbohydrates are polyhydroxyaldehyde are ketones. This is biochemical definition of the carbohydrates, mean that these are the compounds having polyhydroxyaldehyde or ketones are substances that yield such compounds on hydrolysis. Mean that if we are not able to observe aldehyde and ketones immediately, but during their hydrolysis, they produce the uh, products which are having aldehyde or ketones. So many but not all carbohydrates have the empirical formula CH2O whole N, mean that repeated units. So as we see that carbon is bonded with water or attached with water or having water, so that's why they're also known as hydrated carbons. So out of these, there are three main categories: monosaccharides, ready form of energy. Monosaccharides are simple sugars. In living organism, they are having backbone of 3 to 7 atoms. And most monosaccharides of organisms have 6 carbon hexose. Glucose and fructose are hexoses, but are isomers of one another. Each has the formula C6H12O6, but they differ in arrangement of atoms. Glucose is found in blood of animals. It is a source of biochemical energy, ATP, biological currency in nearly all organisms, starting from bacteria till humans, and only not uh, rightly present by the viruses, although when they control the machinery of their host, so ATP will work for rest of the activities. So ribose and deoxyribose are 5-carbon sugar. They are generally pentoses. They contribute to the backbone of RNA and DNA respectively. Disaccharides, they are having varied functions. For example, disaccharide contain, as disaccharides contain two monosaccharides joined by hydrated reactions. So the glycosidic bond is the what we say bond between two monosaccharides. So we'll discuss the detail of carbohydrates in next lesson as well. So maltose is composed of two glucose units molecules it forms in the digestive tract of human during starch digestion. Sucrose, the table sugar. When we name table sugar, it means we name sucrose as the compound. Is composed of glucose and fructose. It is used to sweeten food for human consumption. If we talk about table sugar, so lactose is composed of lactose and glucose and naturally present in milk, mammal's milk. So then comes the polysaccharide, which are energy storage molecules as well as structural molecules. So polysaccharides are polymers of monosaccharides. They are not soluble in water and do not pass through the plasma membrane of the cell. So starch found in many plants in a straight chain of glucose molecule with relatively few side chain branches. Amylose and amylopectin are the two forms of starch found in plants. Glycogen is highly branched polymer of glucose with many side branches. It is a storage form of glucose in animals. Then the polysaccharide, which are structural in nature. So cellulose is the polymer of glucose, which forms microfibrils, that primary constituent of plant cell wall. Cotton is nearly pure cellulose. Cellulose is indigestible in humans due to unique bond between the glucose molecule, because that bond is beta bond, not alpha bond, as compared to the starch and glycogen. So grazing animals can digest cellulose due to special stomach, because their stomach is rumen, complex stomach having different portions in which bacteria are residing there, are naturally present over there as symbionts. So they have those enzymes to digest the cellulose for the animal, and also getting benefit out of that activity. Cellulose is the most abundant organic molecule on Earth, obviously, because cell wall is having cellulose, and plants are numerous than the animals, and structure-wise as well. Chitin is the polymer of glucose with an amino group attached to each glucose. Chitin is the primary constituent of exoskeleton of the arthropods and related, for example, crabs and related animals, lobsters, insects, etc. Chitin is not digestible by humans because the complex bonding nature in it. Peptidoglycan is a polymer of glucose derivatives and found in cell wall of bacteria and are bacteria, yes. Lipid, then comes the lipid, second category. Lipids are chemically heterogeneous group of compounds that are insoluble in water but soluble in non-polar solvents such as hexane, chloroform. So out of the macromolecules, lipids are those molecules which are not polymers of certain monomers. So they are having different combination of their constituent molecules. So, but the chemicals which are what we say insoluble in water and having complex nature, they are lipids. So lipids occur in plants and animals as storage and structural compounds. Storage fats occur in milk and adipose tissue of farm animals and in seed oils. Fats supply over twice as much energy per unit weight as protein or carbohydrates because they are having more CH bonds in them. So lipids are anhydrous due to non-polar nature and represent more energy, yes, giving more energy than carbohydrates, which are readily available form of energy. So lipids are varied in structure. Lipids are hydrocarbons that are insoluble in water because they lack polar groups. Fats provide insulation and energy storage in animals. Phospholipids form plasma membrane, and steroids are important cell messengers. Waxes have protective function in many organisms and also present as the outer coating of leaves. So, out of the lipids, triglycerides, long-term energy storage molecules, fats and oils contain two molecule unit glycerol and fatty acids. So they constitute triglycerides. A fatty acid is a long hydrocarbon chain with a carboxylic acid at one end. So mostly fatty acids in cell, they are of 16 to 18 carbon atoms per molecule, may be saturated, may be unsaturated. Saturated fatty acids have no double bonds between their carbon atoms, unsaturated having double bonds between them, at least one double bond, maybe having more. Glycerol is water-soluble compound with three hydroxyl group. It's an alcohol. Triglycerides are glycerols joined to three fatty acids by dehydration reaction. Fats contain saturated fatty acids and are solid at room temperature. For example, butter. Oils contain unsaturated fatty acids and liquid at room temperature. So animals use fat rather than glycogen for long-term energy storage. For example, the hibernating animals, because it's a good source of energy. Phospholipids, membrane components, phospholipids are constructed like neutral fats, except that their third fatty acid chain along with the glycerol, attached with glycerol, is replaced by phosphate group. And that phosphate group usually having bond with some other molecule like choline, etc. So it is the R group. The hydrocarbon chain of fatty acids become non-polar. Hydrophobic tails, phospholipids arrange themselves in a double layer in water, so the polar heads face towards water molecules and non-polar face towards one another away from the water molecules. So this is unique feature of phospholipids, very important for cell membrane constitutions. Uh, this property enables phospholipids to form an interface or separation between two solutions. For example, anterior and exterior of the cell, the plasma membrane is a phospholipid bilayer. Steroids, four fused rings. Generally, they are having four fused rings. Steroids have skeleton of four fused carbon rings and vary according to attached functional groups. These functional groups determine the biological function of various steroid molecules. Cholesterol is one of the steroid, is a component of an animal cell plasma membrane, and it is a precursor of steroid hormones, aldosterone, testosterone, estrogen, and calcitriol, etc. A diet high in saturated fats and cholesterol can lead to circulatory disorders, uh, although a little bit amount of cholesterol is important for us. So then comes the waxes. Waxes are long chain fatty acids bonded to long chain alcohols. So both the things are different. Alcohols are different in this case. They are long chain, and fatty acid, they are long chain than the triglycerides. Waxes have a high melting point, are waterproof and resistant to degradation by certain microorganisms as well. Waxes form a protective covering in plants and retard water loss in leaves and fruits. In animals, waxes maintain animal skin and fur, trap dust and dirt, and form the honeycomb is an example of wax. Then comes the proteins. Proteins are polymeric compounds made up of monomers called amino acids. So these are large, complex organic molecules composed of one or more long chain of amino acids folded into specific three-dimensional shapes. Protein perform vital biological functions, including catalyzing biological reactions and work as enzymes, providing structural support, transporting molecules, and regulating cellular processes. So a lot of functions are associated with proteins. Protein function metabolic enzymes. Uh, they are the proteins act as organic catalysts to accelerate chemical reactions within the cells. They also provide certain proteins also provide support. So, for example, keratin, which make up hair and nail, and collagen fibers, which support many of the body structures. For example, ligament, tendon in our body. Transport function include channel and carrier proteins in plasma membrane and hemoglobin and transport transport oxygen present in red blood cells. They also provide defense. So include antibodies that prevent infection, interferons as well. Hormones are regulatory proteins that influence the metabolism of cells. For example, insulin regulate glucose content of blood and within the cells. So its function is to regulate the blood, the glucose in between blood and the body cells. So motion with cells within the cells by muscular contraction because muscles are having different components consist of myosin and actin and along with other proteins as well. So proteins are made up of amino acids. So peptide bond is a covalent bond between two amino acids and continuity of polypeptide chain. Atoms of peptide bond share electrons unevenly. So oxygen is more electronegative in that case than the nitrogen. So there will be what we say polarity in the molecule and hydrogen bonding will be possible. The polarity of peptide bond permits hydrogen bonding between different amino acids in a polypeptide chain, not only within one polypeptide chain, but also with different polypeptide chains of a bigger protein. A peptide is two or more amino acids bonded together is a bond. Polypeptides are chains of many amino acids joined by peptide bonds. A protein may contain more than one polypeptide chain. It can thus have a very large number of amino acids. The three-dimensional shape of a protein is critical. An abnormal sequence will have the wrong shape and will not perform proper function. So, Frederick Sanger determined the first protein sequence of hormone insulin in 1953, and this is very what we say important work at that time, and one of the pioneer protein sequence in any biological molecule. So amino acids, they are protein monomers. Amino acids contain an acidic group and amino group. Acidic group is carboxylic group. Amino group differ according to their particular R groups, ranging from single hydrogen to complex ring compounds. R group of amino acid, cysteine, ends with a sulfhydryl group, SH, that serves as connect one chain of amino acid to another by disulfide bond. So this is responsible for covalent bonding between two polypeptide chains. This is very important and meaningful. There are 20 different amino acids commonly found in cells. So then comes the shape of proteins. Proteins shape determine the function of the protein in our organisms. Proteins can serve up to 4 levels of structure, but not all proteins have 4 levels, not required as well. The primary structure is the proteins on particular sequence of amino acid. So tell us about only the sequence of amino acids is the primary structure. Just as the English alphabet contains 26 letters, 20 amino acids can join to form a huge variety of words, mean polypeptides. Secondary structure result when a polypeptide coils are folded in a particular way. For example, alpha helix was the first pattern discovered in a polypeptide bond. Oxygen is particularly negative, hydrogen is obviously positive. So this allows for hydrogen bonding between uh, carbonyl carbon of one amino acid and the NH of another mean carboxyl group and amino group of other amino acids. Hydrogen bonding between every fourth amino acid hold the spiral shape of the alpha helix. The beta pleated and beta sheets was the second pattern discovered. Pleated beta sheets polypeptide turn back upon themselves. Hydrogen bonding occur between extended lengths. So these are the bondings. Then comes the fibrous proteins. For example, keratin are structural proteins with helices and pleated sheets that hydrogen bond to one another. Tertiary structure result when proteins are folded, giving rise to final three-dimensional structure of proteins. This is due to interaction among R groups. The secondary structure due to the hydrogen bonding between carboxylic and amino groups, but tertiary structure most of the time is due to the interaction, maybe in hydrogen bonding, maybe any other due to R groups of the constituent amino acids. Globular proteins tend to ball up into rounded shape, mean they will attain globular shape, rounded shape. Strong disulfide linkage maintain the tertiary shape. Hydrogen ionic and covalent bonds and interactions are there. Hydrophobic interactions even. We will discuss it later on in protein lesson. Quaternary structure result when two or more polypeptide chain combines. So for the sake of quaternary structure, we need at least two polypeptides, maybe more. Hemoglobin is globular protein with quaternary structure of four polypeptide. Each polypeptide has a primary, secondary, and tertiary structure in hemoglobin or any polypeptide, any molecule with more than one polypeptide chain. So then comes the nucleic acids. Nucleic acids are large biomolecules composed of chains of nucleotide monomers. So out of biomolecules, carbohydrates, proteins, and nucleic acids are polymeric molecules. But lipids are not polymeric most of the time, although having combination of many constituent molecules, but they are not having same repeated units in different forms. So they store and the nucleic acid, they store and transmit genetic information essential for life. The main types are DNA, which carries heredity material. Sometimes RNA is also heredity material in some viruses, and RNA, which play key role in protein synthesis and performing the gene regulation function. Nucleotides, the building blocks of nucleic acid, each consist of a phosphorus group, a sugar, either ribose in RNA or deoxyribose in DNA, and a nitrogen base. The nitrogenous bases are classified into two types: pyrimidines, single ring, 6 carbon, and purines, double ring, 9 carbon molecule, 9 ring rather, not carbon. So key points about nucleic acid. Nucleic acids are polymers of nucleotides that perform highly specific function within cell. DNA, deoxyribo nucleic acid stores the genetic information necessary for its own replication and for determining the amino acid sequence in proteins. So this is important because proteins are needed, and rest of the body function and living cells function, they may be performed by the proteins. So as we know, that central dogma of life is about DNA, RNA, and protein. So RNA, ribonucleic acid, enables the translation of the genetic code in DNA into amino acid sequence of proteins and also perform various regulatory and catalytic role in the cells. We talk about RNA. Some nucleotides have important independent function as well. For example, they may work as coenzymes. They are nucleotide-derived molecules that assist enzymes in catalyzing biochemical reactions. ATP, well-known molecule, and also acting as ATP biological currency. So adenosine triphosphate is a nucleotide that serves as a primary energy currency of the cell, providing energy for biosynthetic processes and other energy demanding activities. So structure of DNA and RNA, nucleotides are the molecules complex of three types of molecules: a phosphate, phosphoric group, pentose sugar, and nitrogen containing base. So DNA and RNA differ in following ways. A nucleotide of DNA contains deoxyribose sugar, and nucleotide in RNA contains the ribose sugar, pentose sugar. In RNA the base uracil occurs instead of the base thymine. Both RNA and DNA contain adenine, guanine, and cytosine. DNA is double-stranded with complementary base pairing. RNA is single-stranded. So, complementary base pairing occurs when two strands of DNA are held together by hydrogen bonds between purine and pyrimidine base on the other strands, opposite strands. The number of purine bases always equal and number of pyrimidine bases. In DNA, thymine always paired with adenine, cytosine always paired with guanine. Thus, DNA is having A plus G, and this A plus G combination should be equal to C plus T combination. Two strands of DNA twist to form double helix. RNA does not form helix, only can be folded back if the longer RNA molecule is present. So ATP, adenosine triphosphate, ATP is a nucleotide in which adenosine is composed of ribose and adenine. So not deoxyribose. So triphosphate derived their name from three phosphate groups attached together at the end of the ribose, tri, di, mono. ATP is a high energy molecule because last two phosphate bond release energy when broken. So this bond is energy rich molecule. In cells, the terminal phosphate bond is hydrolyzed, having and leaving ADP molecule, and energy is released. This is easiest way. Otherwise, the ADP may also hydrolyze into AMP, adenosine monophosphate. The energy released from ATP breakdown is used for the all energy requiring processes of the cell, such as synthetic reaction, muscle contraction, transmission of nerve impulse, etc., and different other functions. So then comes the vitamins, are small organic molecules essential for various biochemical processes in the body. They often act as coenzyme, helping enzyme function properly, and are acquired in small amounts through diet, because body cannot synthesize enough of them. We'll talk about animals. So animals need vitamins for their general metabolism. Hormones are biomolecules, are chemical messengers. They work as chemical messengers produced by glands in the endocrine system. They travel through the bloodstream to target organs where they regulate physiological processes like growth, metabolism, reproduction, mood, and many other functions. So that is about biological classification and a little bit function of biological molecules. And next of the lessons, we will discuss the precise topics which are given in your syllabus. Till then, Allah Hafiz.